Thank you so much for that lovely introduction, Maria and Jan. And thank you to the organizers, Sherry and David, for inviting me. This is a huge honor to be invited down here. It's a long trip from North Carolina, but it's well worth it. It's a beautiful country you have here, and I'm always delighted to visit. Keep me on your schedule. So let's see if I can make everything work here. I get to present to you, let's see if I have a pointer. Where'd my pointer go? There it is. Okay, thank you. Sorry about that. All right, so I get to talk about something that I've been working on for the past 25 years, and that is bacterial extracellular vesicles. And unlike perhaps many of you here, I actually chose to work on EVs. They were a really exciting opportunity I found. When I was beginning my laboratory, I worked on yeast intracellular vesicles, COP2 vesicles in Dr. Sheckman's lab, and I had heard that bacteria make vesicles, extracellular vesicles. And back in the dusty archives at Berkeley, I was looking at these old plates, images of bacteria making vesicles, particularly Neisseria gonorrhea, Neisseria meningitidis, and Vibrio cholera, and even E. coli. And this was really fascinating to me, and I wondered how this happened. And that's how I began this journey. And we can appreciate that bacteria live everywhere. Oftentimes we only think of the human host and pathogens, but really they're all around us, and thus the bacterial vesicles that they secrete are also all around us. So two overarching questions have guided our research over the years. How are bacterial vesicles made, and why are bacterial vesicles made? This seems like an enormous expenditure of molecular energy for a cell. But first, why do we care? Quantitative and mechanistic studies on bacterial EVs can help elucidate general biological questions, understanding basic physiology of bacteria, novel mechanisms of interspecies and interkingdom communication, new insight into antibiotic resistance mechanisms guiding EV cargo selectivity and production yield. Also, of course, translational applications. By interfering perhaps with the transmission of virulence factors and inflammatory mediators, identifying markers for infection diagnostics, aiding EV-based vaccine development, and by improving development of targeted nanoparticle therapeutics. So there's many, many reasons, whether you're a general biologist or more interested in translational medicine, to study bacterial EVs. So again, back to our overarching questions. How these bacterial vesicles are made. Is it purposeful? Is it selective? How is it selective? What genes or gene products are involved? That was the question Randy asked when I left his lab. Why are they made? Are they beneficial to the producing cell? And what and how do they communicate to neighboring cells? And I hope to answer some of these questions with you today. So the biochemical, physical, and genetic data support the concept that bacterial EVs are not lysed cell debris. And I spent many years initially proving this concept to essentially very dubious critical reviewers. So what I found was BEVs bud from intact cells, and I'll show you the data that they differ from cells based on their zeta potential. They show selectivity of cargo, both protein and lipid, and their specific genetic factors that govern their biogenesis. So first of all, here is an atomic force microscopy image that shows E. coli, just lab strain DH5 alpha, growing at 20 degrees. These are live cell images of E. coli budding a vesicle. And this was taken in collaboration with Claretta Sullivan, and you can see here that there is a vesicle that's budding from the surface and leaving behind an intact cell after the vesicle pops off. So that was exciting for me to see, because pictures are worth 1,000 experiments sometimes. And here is an experiment that we did with enterotoxigenic E. coli, a pathogenic form of E. coli. In the inoculum from the in vitro grown culture, by scanning electron microscopy, these bacteria look kind of smooth. About 90% of them have this phenotype. However, after 2 hours, after being inoculated orally into the mouse and recovered from the intestine, these bacteria have these bumpy vesicle-like features on them. And these are not disintegrated lysed bacteria, these are growing, dividing bacteria. So 84% of the bacteria that we investigated, that we detected, were looking like this. So then, to further investigate bacterial EVs, we need an isolation protocol. And so I developed this protocol way back when to isolate bacterial vesicles from cultures in vitro. And it's a standard procedure that now I think a lot of people use. You grow your cultures, spin down the pellet, concentrate those vesicles. Either you might use tangential flow if it's a high volume, and take the retentate and filter sterilize that. Bacteria are small, so you need a small filter size. And then ultracentrifuge that and get a crude bacterial EV pellet, which you can use for yield determinations. For isolating purified vesicles, I developed a density gradient protocol, again, one that is used widely today, using OptiPrep or iodoxanol, in which the bacterial vesicles float in an osmotically appropriate environment. And then the fractions could be identified for which contained the BEVs. And the only way I really had initially to look for these, I'd never purified these before until I started this project, was to look for round things by EM, essentially, and for outer membrane proteins, which I expected to find in the fractions by STS page analysis. And so you can combine the fractions enriched in those EVs and store them and analyze them. And then we took pictures of them here by transmission EM, and you can see a range of diameters of 50 to 250 nanometers, approximately heterogeneous in size, nothing like the COP2 coated vesicles I'd worked with as a postdoc. They could be quantitated by protein amounts using Bradford or lipid diintercalation FM464, which I found was useful for these vesicles. And their yield is often normalized to bacterial counts by colony forming units or by OD. So bacterial vesicles contain select envelope cargo. They contain a subset of envelope components, notably for outer membrane vesicles, which are derived from gram-negative bacteria, which have two membranes, the outer membrane and inner membrane. The outer membrane vesicles that bud off of the outer membrane contain lipopolysaccharide, the external lipid of gram-negative bacteria, as well as outer membrane porins and proteins that are embedded in the membrane, as well as periplasmic proteins in the lumen, so the soluble content of these vesicles. And all bacterial vesicles, also from gram-positive cells, contain phospholipids. In this case, for OMVs, it's the interior lipid layer of the outer membrane, peptidic glycan and fragments, probably thereof, surface-associated proteins, which now have a name, the corona, and nucleic acids, RNA and DNA can be found in these vesicles, small molecules and metabolites as well. So we were curious recently whether we could demonstrate the selectivity of surface cargo using zeta potential. And I won't go into the physics of it all. You'll hear presentations that are much more in depth about this later on, I'm sure, by the fabulous instrumentation representatives here. But suffice it to say that the zeta potential is the electrical potential at the slipping plane in which the charges are not fixed on the particle surface. So they're not actually a direct, it's not a direct reflection of the charge of the particle itself, but it is at a distance from the actual charge of the particle, but it does reflect properties of the surface of that particle. So we thought we'd use zeta potential to investigate vesicles versus cells. And so in this data, we actually also looked at yeast vesicles, and here are data from gram-positive and gram-negative bacteria, Staph aureus and Pseudomonas fluorescens. We looked at cell zeta potential in blue versus the vesicles which are derived from those cells that are shown here in yellow. And you can see that the vesicles have a different zeta potential measured at different pHs. The ones for gram-positive bacteria are less negatively charged than the cell, And for gram-negative bacteria, the cell is more negatively charged than the vesicles. And so this indicated that there is selection of some surface components. So what are these? Well, proteins are selectively incorporated into vesicles. You can see this by a simple comparison of the protein profiles from the membrane, the outer membrane in this case of Pseudomonas aeruginosa, does not resemble the composition of the outer membrane proteins present in these vesicles. So they're similar but not identical to the membrane of origin. And their composition depends on the species, the strain, the conditions that they were grown in, etc. So we studied the mechanism of protein selectivity into bacterial EVs in E. coli and enterotoxigenic E. coli. And I'll just take you through a quick summary of this paper since I talked about it last ISA. In this case, we engineered versions of the same protein A in an untethered and a tethered version. So the tethered version is actually non-covalently attached to the cell wall, the peptidoglycan here in the periplasm, and the untethered version doesn't have that piece of that length of protein, so it is not tethered to the cell wall and it is free in the membrane. It is an integral membrane protein, however. And what we found was that the vesicle to outer membrane ratio of the tethered version of this protein was much less than the untethered version. So the untethered version was free to leave in the vesicles, as you might expect. Actually, under oxidative stress conditions, that ratio increased, which was interesting because what this was all based on was some experiments we had done with enterotoxigenic E. coli in which we found that under unstressed versus stressed, oxidatively stressed conditions, the composition of the vesicles and the ratio of proteins changed to that was present in the membrane. And so this was consistent, this model system, with what we saw sort of in real life with E-Tech. And so the membrane-only proteins that were not tethered to the cell wall were more free to leave in the vesicles. And under stress conditions, actually, we determined that the cells must be making these large envelope complexes, trans-envelope complexes, and those are not available for departing in these vesicles. So there's some mechanistic basis for having selectivity of cargo in those vesicles under different conditions. So what about lipid selectivity? So this takes us to our hypothesis that outer membrane vesicles enable outer membrane remodeling. And this comes actually back to the observation that Neisseria makes so many vesicles. Well, Neisseria species are really good at remodeling their membrane. If you think about how they survive in the body and cause infection, they're very good at changing their coat, their outer membrane. But we chose Salmonella as a model system to study this because it's so well described. So we posited that there would be enrichment of the old type of LPS as the cell is remodeling its membrane for this new environmental condition. And this would be a means by which the bacteria could remodel and change its coat. So it would just throw out the sort of old material. So Salmonella has a well-adapted intracellular lifestyle. It enters from the intestinal lumen, hopefully not currently in any of your lumen right now. It'll cause great distress. But it enters into the salmonella-containing vacuole, which is an acidified compartment. It has low pH, low magnesium concentrations. The salmonella replicate there, and then they exit into the submucosa back to neutral conditions. And this process by which the bacteria adapt to this very acidic condition has been well studied, and that's why we chose to use it in our model system. So essentially, all you need to know is that the lipid on the exterior surface changes. It changes from an unmodified species to a modified species. And I'll show you more details in a moment. But this is our experimental setup that Catherine performed in the lab. We took Salmonella at neutral conditions, high pH, high magnesium, shifted them to low pH, low magnesium, or Mach shifted them to the original conditions, and then collected vesicles during this process of shift. And the bacteria will change their coat. In this case under these acidic and low magnesium conditions, and not in these in the Mach shift conditions. And what you can see by thin layer chromatography of the lipid A, the LPS, over time is that the LPS species change. You can appreciate that. And these are examples of the highly modified LPS that the cell changes to accommodate life in this new acidic condition. And we can analyze this by mass spectroscopy. I don't have time to go into it. This is the outer membrane species. Originally, these are unmodified, and then they shift. After 2 hours, the LPS changes in character. It becomes much more modified. So here's that same time course of the outer membrane. The LPS is shifting to these more complex bands in TLC. And then we isolated vesicles at the conditions at 30 minutes after shift and 60 minutes after shift. And you can appreciate that the vesicle bands are different than the cells at those particular time points, right? So these are more unmodified species compared to the cells at those time points. So then we can quantitate that, and to do this so that I could understand it, Catherine color-coded it for me, so that these blue bands are sort of the unmodified species. So as time goes on, there's less and less of the unmodified species, and these brighter colors are the more ratios of the more highly modified LPS species. So they're coming in over time. And then here is the composition of the vesicles for those species. And you can see that those very modified species are not present in the vesicles, even at 90 minutes. And so we can do this. This is the empirical data. This is what we got. But you can also theoretically calculate what should the vesicle composition be if there was just stochastic incorporation of the lipids that were present in the cells at that time. And so that's what we did. We compared theoretical and observed for the Mach shift conditions, which are essentially the same, and the shifted conditions. And what was interesting here, just to point out, is this difference. There are far fewer of these modified species in our observed than should have been present had there just been stochastic incorporation of lipids into those vesicles. But what was actually a surprise was the shift back. So now we're thinking about the vesicle, the salmonella that are contained in this acidic compartment that are now released back into neutral conditions and high magnesium conditions. And so we thought this was a control, but it isn't. So at this point, the bacteria are have highly modified LPS. So that's at time 0. They're highly modified LPS. They've been growing in acidic conditions for a long time, and they're just growing happily. They have all this modified LPS. And over time, we shifted them to these neutral conditions, as would occur maybe physiologically. And they sort of change back to unmodified species of LPS, right? So these darker blue colors come back in. And then we collected the vesicles again during this shift. And what was shocking, even in our Mach shifted conditions, that the theoretical and the observed conditions were not what we expected. So theoretically, we should have had all these highly modified species present in the vesicles. These are not even cells that are shifting conditions at all. And yet, the observed vesicle lipid composition was highly unmodified. So the cells are retaining this modified LPS at all costs. They're finding islands, if you will, of unmodified species and chucking them out into vesicles. So there's something perhaps physical about what is actually entering the vesicles. And so in this shifted condition to the neutral conditions, we see the same thing, where there are much more unmodified species in the vesicles rather than modified species. So we tried our best to see if we could think of the rules of lipid incorporation under these conditions, what we came up with was perhaps a polymorphic regulation of the lipid A composition as far as what is getting expelled in outer membrane vesicles based on the head group charge and the hydrocarbon length versus what is retained in the cell. And you can read more about that in Catherine's paper from some years ago. So now I wanted to move on to the question of how this can occur without cell lysis. right? So these intact bacteria, how can they produce these vesicles? And mechanistically, how are they doing this? And I'll tell you about a couple of mechanisms that we've studied thus far. Periplasmic crowding and cell wall outer membrane, what I think of as staples, as well as the corona in action. So for the first couple of stories, I'll bring you to a genetic mutant screen that we did. We isolated vesicles from all 4,000 single gene mutation knockouts of E. coli K12 strain in 96 well plate tiny cultures. And we performed this assay that Adam Culp developed in the lab, whereby we grow these tiny cultures and filter them through a 0.45 micron filter, take the resulting cell-free supernatant and deposit that onto a nitrocellulose membrane, so a little tiny Western blot membrane, essentially with little spots on it, and then we immunoblotted that for anti-LPS. So it's a crude determination, for sure, for what is a vesicle. But LPS is in all of our vesicles, so it seemed to be a good marker for us. And then we could do densitometry and relate that to how well the culture grew, et cetera. We got rid of poor growing or lysed mutants so that they wouldn't confound our screen. And we then determined a vesiculation score. How many vesicles were made per how many bacteria were present in that culture. And so you get your typical curve of generic mutations here that made not much of a difference in phenotype, but then there were those that had greater than 2 standard deviations from the wild type amount of vesicles. So hypovesiculating mutants down here, hypervesiculating mutants up here. And I'm just focusing today on studies that I'll summarize about 2 of these mutants, the DEG-P mutant and NLPI mutant, that taught us something about vesicle biogenesis. And that's all summarized, many years of work in this slide. So DEG-P is a protease chaperone. It exists in the periplasm. And in the absence of DEG-P, periplasmic misfolded proteins accumulate, particularly when the cell is stressed. So think of a stressed cell, it accumulates misfolded proteins, and DEG-P is there to clean up the mess, either to help refold it and bring it back to happy conditions, or degrade it if it's hopeless. And what happens in the absence of DEG-P is that the cell just accumulates, sort of constipated with all of these misfolded proteins. And the vesicles end up being a release valve, a garbage dump, if you will, for those components. So it can release this extra material extracellularly. And this process is, in fact, important for growth. So if you actually add under a hypovesiculating mutation to a DEG-P strain and stress it, it will not divide. So this is an important, critical process for bacterial physiology. And secondly, we learned something about from NLPI, which is an outer membrane anchored lipoprotein, not the lipoproteins you're used to thinking that contaminate your preps, but the lipoproteins that are literally lipid anchored into the membrane. So this is anchored into the outer membrane here. And NLPI, we found through a series of genetics and biochemical experiments, is responsible via SPR for the regulating the peptidoglycan biosynthesis and degradation. So peptidoglycan, the cell wall, is constantly being remodeled in bacteria, and NLPI kind of puts the brake on this, this process. So in the absence of NLPI, the peptidoglycan is just recycling and cycling and cycling constantly. And it cannot establish these staples, which are covalently linked proteins that are lipid anchored, again, another lipoprotein that's anchored in the outer membrane and covalently attached to the cell wall. And it just loses the ability to make those staples. And as a result, we think vesicles are overproduced. So in this case, outer membrane vesicles are a supplemental envelope stress reduction pathway, and in this case, outer membrane vesicle biogenesis is regulated by envelope linkages. There's a lot more to be learned from our list of hypo and hypervesiculators, and many graduate student years of work ahead of us to understand why these mutants have these phenotypes. So I wanted to also tell you about the corona in action. The corona has gotten a lot of press lately. It has a great name. I'm happy to hear that. It used to be, we just called it protein that was bound to the surface of our vesicles. But now it has a cool, fancy name, corona. I love it. I wanted to tell you that it isn't just stickiness. It isn't just the fact that it's a protein that happens to co-migrate with your vesicle prep. In this case, I'll tell you the story about enterotoxigenic E. coli heat label enterotoxin. So it secretes this toxin, LT, which is an AB5 toxin. It's an A catalytic subunit, pentameric B subunit as the binding to eukaryotic cells. So that's what enters cells and gives people, unfortunately, traveler's diarrhea. So if you drink the water that's contaminated with ETEC, you're in for a bad week, but it does cause dehydration deaths in the millions per year, so it's a very serious illness. So what was found by many groups is the binding sites of LT, this toxin for GM1, its mammalian cell receptor, and blood sugar antigen, which is interesting. It binds a lot of sugars. It binds blood sugar antigen as well. But we found it also binds lipopolysaccharide, and we did many studies to show exactly where that binding site is. It's independent of both those other binding sites. But the discovery of LT as a corona on ETEC vesicles came about very early in our studies, because I was really curious how this LT was secreted. At that time, LT was thought to be only periplasmic, so I was confident it would be only in the lumen of the vesicles. But instead, what we found is that there's a secretory pathway that exports the LT out of the cell, and we found it was associated with the vesicle surface, and finally we also found that it was in fact associated with the cell surface. So LT is secreted, LT binds LPS. We studied the molecular details of that aspect for many years, and this corona of LT binds GM1 on mammalian epithelial cells, conferring toxicity into those cells. So it's this exterior corona of LT that leads the vesicle and brings the vesicle into the mammalian cell via receptor-mediated endocytosis. And what we found was that this LT was releasable by EDTA, and that the toxicity was also susceptible to proteolytic degradation, external proteases, as well as external application of GM1. So this corona of LT exists on these E. coli vesicles, these ETEC vesicles. But recently, we considered whether LT could also potentially function in vesicle biogenesis. And our hypothesis is that LT might, by its ability to bind LPS, increase production of vesicle by altering these membrane properties right there. And the reason we thought this was that in a mutant of ETEC that cannot produce the toxin, we have much less yield of vesicles. ETEC normally produces a very high number of vesicles, and LT reduced this the lack of LT reduced this considerably. And the lack of being able to secrete LT also reduced this considerably. So this cell, this ETEC cell, makes LT, but it cannot secrete it. So that indicated that LT externally was doing something to increase vesicle production. So we then put in a plasmid, we transformed that strain that isn't expressing LT with an inducible plasmid, so it is expressing LT under IPTG control, under inducing control, and we found that we could increase vesicle production. And we have an LT mutant, again, from years of study that we knew could not bind to LPS. And so this T47A was expressed in these cells, and that did not cause an increase in yield of the vesicles. So that was very intriguing. So we looked at that exact interaction. This is the isothermal titration calorimetry to show that LTB, the B subunit, is binding to LPS with micromolar affinity. And we tested, since we're working with E. coli, all these different forms of E. coli LPS to see which type of LPS the LT prefers and which it binds best. And so we performed all of these co-sedimentation assays to look at LT binding to LPS. And this is in order of preferential binding from most binding to least amount of binding. And we decided that LTB binding requires a terminal galactose moiety. And then we looked here by FTIR to see what was occurring exactly when LT was binding LPS. And essentially what you need to take away here is that the LTB binding is immobilizing the phosphates and the sugar residues upon binding. It's stiffening that LPS. And we could see that by looking at the heat capacity of the LPS as well. I don't have time to go into all these experimental details, but suffice it to say that the binding of LT increases the enthalpy, increases the stiffness of the LPS in its phase transition. So we think that this LT binding to, as a corona protein, binding to the lipid in a very specific way is changing the properties of the membrane when it's binding. And as a result, there's a physiological effect of increased vesicle production. All right, so finally, I'm going to end with a functional story. And I'm looking at this perhaps in a view that's a little different than the perspective that you're used to. We're looking at a significant model system that oftentimes is ignored, and that is the plants, the ecological system, the environmental world around us. What do the vesicles do in plant biology? And so I had a student, Hannah, who was really interested in looking at this, and she isolated vesicles from a plant pathogen, Pseudomonas syringae, and a plant, what she considers a commensal, Pseudomonas fluorescens. And we isolated these under complete and minimal media conditions. We could isolate vesicles, we could characterize their protein amounts, their lipid amounts, their sizes by EM, and they were not different in any notable sense. But we had all these preparations of vesicles that we could then use to investigate their function. And we know from mammalian studies that vesicles are very good at delivering antigens into mammalian systems and eliciting a protective response, i.e. a vaccine type response. So what about in plants? So that's what Hannah tested. She used a challenge with the Pseudomonas syringae, the pathogen, which yellows the leaves. So if you just leave the leaves untreated, they become yellow when you infect them with this Pseudomonas syringae. But hopefully, you can appreciate the color here. If you pre-treat with vesicles from the syringae, if you pre-treat that leaf with those vesicles and then challenge it with this pathogen, the leaf does not yellow. If you do this actually with the commensal vesicles, the leaf doesn't yellow. So both of the PST and the fluorescence vesicles are able to protect plants from infection. And this is a dose-dependent process. The vesicles were applied at different dosages and had appropriate dose-dependent response. And what was interesting, we had a freezer full of vesicles from all kinds of other studies we've been doing over the years. And so we investigated, is it just specific to Pseudomonas vesicles, that this protective response? So here's the protective response of syringae. So there's fewer of the challenged bacteria growing on those leaves if we pre-incubate with the syringae vesicles. Its close cousin Pseudomonas aeruginosa vesicles wasn't that protective, interesting, but enterotoxigenic E. coli vesicles were protective. So that's particularly fascinating. Staph aureus vesicles were not protective in this system. So there's certainly a lot we don't know about why these vesicles are protective in this plant scenario. We're doing omics these days to look at the differential cargo in these different vesicles between the different types of preps that we have to see if we can identify what in those vesicles might be responsible for the protection. And so I will leave you with the acknowledgement of a great group of students and postdocs over the years whose work I highlighted today: Hannah, Clarice, Nicole, Caitlin, and particularly Catherine, and Amanda and Adam, who did some of that early work, as well as Carmen, Ben on his work on LT. And our collaborators at Duke and elsewhere, as well as our funding sources. And I appreciate your attention. Thank you.